Share screen, start the timer. Okay, now start timer. Now start the timer. Okay, cool. Get into it. Okay, cool. Perfect. Hello, class. My name is Eric Carrera, and today I'll be discussing a little bit more about the nurse sharks. Yeah, let's kind of get into it. So, the title is The Mystery of the Nurse Sharks by me, Eric Carrera. Uh, the table of contents. So, the next slide is table of contents. So, on slide 3 and 4, I'll discuss a little bit about the evolution of the nurse sharks. On slide 5, I'll discuss the environment. And on 6, I'll talk about the nurse shark's immune system. And on slide 7, I'll talk about DNA analysis. So yeah. So the first slide, the evolution of the nurse shark. Nurse sharks, aka cat sharks, aka bottom feeders, first appeared around 56 million years ago. Many scientists, or that's what scientists believe, because they can't essentially, pinpoint the exact same date or year they came from. But after analyzing, their DNA analysis, not their DNA, sorry, their fossils, after analyzing them, they believe that they first came around 56 million years ago, so that's pretty cool. And the nurse sharks, they were around the periods of the Paleogene period, the Neogene period, and the present, which is the Quaternary period. And these little guys, or these nurse sharks, weren't first written about or documented until 1788 by a man named Joseph Bonateri. When he did essentially did first document these nurse sharks, he had them under the family of the Squaleus ceratos. I think that's how you pronounce it. But this would later be changed in 1841 into the family with their still tagged along with, which is the Ginglimostoma ceratum. Kind of sounds like a Harry Potter spell. They're all funny names. So on to the next. So the evolution of the nurse shark. So many scientists, even though sharks have been around for 400 million years, they believe that all sharks originated from this fish on the bottom left-hand corner called the spinefish or the spiny fish, or its professional name known as the uh, Acanthodian. And this shark would later have this little fish would evolve different sharks, but then we move on to the carpet shark family, and that specific shark in the carpet shark family is the has a weird. They all have weird names. It's the Carcharhini formius, and then that shark would later the nurse shark would later evolve from this shark. But many scientists believe that the nurse shark originated from the carpet shark family or came from the carpet shark family because there is a shark called the zebra shark that has many similarities. So many scientists believe that possibly at one point in time, these two sharks had a common ancestor. So yeah, it's pretty interesting. The environment. If any of you are interested in looking or finding a little more about nurse sharks, you could find them in tropical and subtropical coastal waters, specifically in the Atlantic Ocean and the Eastern Pacific. And if you watch any documentaries, you could usually find these nurse sharks and inhabit a lot of reefs. Um, uh, I like to think of these nurse sharks kind of like cats. They do tend to sleep, during the day, sleep, and they're up at night, kind of like cats. So they rest during the day and they prey on night. And at night, when they do go on the prey and they do go on the hunt, they do tend to look for squid, small stingrays, and spiny lobsters. Just to name a few things they do like to eat and hunt. Um, the next point is, do they have predators or a threat? So scientists believe they don't have a threat. There's not a specific like shark on the hunt to go eat these guys, but it has been documented, unfortunately. As you can tell on top of the presentation, you can see a hammerhead shark. Lemon hammerhead sharks, bull sharks, and blue tiger sharks do tend to sometimes eat these little guys, which is unfortunate for them. So yeah, it sucks. On to the next. The next will be the nurse shark's immune system. So when you think of um, nurse sharks, um, or sharks in general, you don't really hear a lot of like sharks in general, or sharks get sick. Have you ever heard of that? Of them being infected by a virus, get a cold, get a flu, or whatnot? You don't. And why is that? And I'll kind of tell you why. So, um, at the time, while I was researching about a little more about nurse sharks, I came across an article written by the Washington Post, and they written about this man on top left named Aaron Lebu. I think that's how I pronounce it. And he was researching at the time to possibly find a cancer cure um, through the antibodies of llamas and alpacas. And while he was doing that research, there's also a Scottish biotechnology company named Ellis Morgan. Morgan. They were also doing the exact same thing, but they were doing it uh, through trying to find that through nurse sharks. And the reason why they wanted to study nurse sharks is because sharks have had a strong immune system that's helped them survive, for more than 400 million years. But how is that possible? So they decided to do study nurse sharks, and I believe they wanted to study nurse sharks because they. I don't think they wanted to study like sharks such as like the tiger, bull, or great white. Because if anybody's ever seen the movie Deep Blue, with I believe, or I think it's called Deep Blue, with L. Cool J, and they study a great white. If you've seen the movie, you know what I'm referring to. It doesn't end well for the scientist or the doctor, or whatever it may be. But um, so the way that they did this study or this test, as you can see in the bottom left and bottom right, they would how they research the nurse sharks is they would look at their antibodies, and what they would do is at the end of every month they would test the nurse sharks. So before the end of the month, they would have, I believe, 3 to 4 nurse sharks between 2 to 4 feet, and they would feed them daily, every day or two, and they would feed them crab, squid, and what else? I think that's it. There might be more, but um, they would do that until the end of the month. At the end of the month, they would look at their antibodies, and they noticed that, compared uh, to human antibodies, shark antibodies are much better at neutralizing invading viruses. When we tend to when the human immune system senses the presence of a foreign cell, it usually releases a certain protein called antibodies, of course. And essentially, what the antibodies do, they grab onto a specific molecule um, on the surface of the virus or bacteria or tumor cells or whatever it may be. And when this happens, they tend to call up support from the potent immune system. When the potent immune system, which are called T cells, and essentially what T cells do, they come in and they kill invaders, smash them off, and which is good. But besides that, scientists have also developed also man-made antibodies, which are injected to the body to also do the exact same thing, fight off foreign cells. When the synthetic antibodies have found their target, like I said, they switch on the immune system to attack foreign cells. But one big difference between human and nurse shark or shark antibodies, um, 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 my apologies, sorry. One big difference between human and man-made antibodies is that they tend to be a lot more bulkier, and they're more Y-shaped. So Y-shaped molecules. So due to their size, they are usually only able to bind to smaller uh, number of targets of invading cells. This explains a lot to scientists on why human and immune system and antibody-based medication is not always the best of fending off enemies, or they're not always 100% to get rid of the virus. But unlike nurse shark, their antibodies are less than one tenth the size of human antibodies. This allows their antibodies to penetrate essentially deeper into the tiny cracks found onto the surface of bacteria or cancer cells or any virus. And any virus. This improves the chances of them grabbing onto the surface of the virus and staying with them, and and having the chance of the immune system destroying the invading cells, which is very, very interesting. And then from this, the antibodies, that company I referred to, Ellis Mungan, they're also currently doing possible um, possible cure for, I believe it was triple negative. I think it's called triple negative breast cancer. So they're currently using these antibodies to see if they can come possibly up with a cure to fight that. And they won't have trials until 5 to 10 years from now. But they also, while Aaron LeBout and this company Ellis Mungan, they were studying their sharks antibodies, they tried also to see if there was a cure for fending off the coronavirus. And guess what? And I think in 2021 they did some testing, and I believe it took down majority of the different variants of viruses, including I believe it's the Omicron um, virus. So that's pretty interesting. And one cool thing I think about what's so interesting about nurse shark antibodies um, is that their antibodies, if they were to be used for medicine, hopefully you never know, 5 to 10 years from now, they don't need to be kept cold, like they do now. And so medicine, if they were to use their antibodies, they could possibly, if they do use them, they could be shipped around the world more easily. So that's that's pretty cool. I think so. So on to the next and final slide. So the biotechnology I chose, which was DNA analysis. What is the purpose of this technique? The purpose of this technique of DNA analysis on nurse shark serves for several purposes. It could help contribute towards, like I said, the study of nurse shark antibodies and have a better understanding of the genetic basis and the diversity of their own repertoire. And how does it work? There are different techniques that revolve around DNA analysis. What are some of the applications of these techniques? I think there's several of them that use DNA analysis, but some of them could be DNA extraction, DNA barcoding, and next generation sequencing, which is NGS. So just a little bit about DNA extraction. Essentially what this is it's just extracting DNA from tissue samples. It could be a fin clips or blood samples. I believe there's a video on it that I believe a scientist was doing or using that. But then for DNA barcoding, essentially what it is, it utilizes specific DNA region, which region where often the COI sequence, which is used as a genetic barcoding for species um, identification by having these um, compared to the COI sequence from the nurse shark um, with the reference circuits in the database. This can help researchers determine the species identity of individuals, or even the very presence of a specific nurse shark species, which I think is pretty cool. And like I talked about, the NGS, the next generation sequencing, which is this technique has been evolutionary for DNA analysis. It has allowed for a higher throughput sequencing of DNA fragments that provide extensive, genomic information, which I think is pretty cool. Next generation sequencing, or NGS, is employed for various application, which includes a whole genome sequencing. I believe the whole genome sequencing, transcriptions, and population genomics. This enables a comprehensive understanding of the nurse shark genomes and gene expression profiles. So I think that was pretty interesting. But yeah, I think that is the last one. So I appreciate you, who's ever here watching, for lecture credit or whatnot. If not, that's fine. If it's just the professor, hello and thank you for listening. And if anybody is watching for lecture credit, I just wanted to say that it's been a pleasure to have class with you guys, and I wish you guys, or I wish you all, nothing but the best, and hope you achieve your goals, whatever that may be. So, yeah, I think that's it. Yeah, these are my works cited, everything that I've used, and I think that is it. So I appreciate you, stopping by and listening to me. So, thank you, and enjoy the rest of your day. Bye now. And stop. Wait, hold on.